Mesdames et Messieurs, good afternoon, ladies and gentlemen. Welcome to another session of CSPC virtual series on COVID-19, and this time in partnership with Genome Canada and a panel titled Lifting the Curtain on CanCoGen. As you may know, that on April 23rd, Prime Minister Justin Trudeau announced $40 million funding for a project called Canadian COVID Genomics Network, CanCoGen project, en français, Réseau canadien de génomique COVID. This project is led by Genome Canada and is in partnership with the 6 regional genome centers, as well as national and provincial public health labs and genome sequencing centers through CGEn, hospitals, universities, and the public sector. The CanCoGen project aims to coordinate and scale up existing genomics-based COVID-19 research in Canada and internationally in order to accelerate public impact. The network is now up and running. Now we would like to lift the curtain and find out how it works from our 4 panelists, or 5 panelists, who will shed the light on the overall project, the virtual sequencing, host sequencing, national data collection, the international collaboration aspects of the work underway. And let me introduce our panelists today. Dr. Catalina Lopez-Correa, the new Executive Director of CanCoGen, Dr. Fiona Brinkman, Distinguished Professor of Bioinformatics and Genomics, Department of Molecular Biology and Biochemistry from Simon Fraser University, Dr. Lisa Strug, Professor of Statistical Science, Faculty of Arts and Science, University of Toronto, and Health Researcher in Genome Data Sciences and Senior Scientist at the Hospital for Sick Children. And Dr. Gary van Domselaar, Chief of Bioinformatics from National Microbiology Laboratory, Public Health Agency of Canada. And last but certainly not the least, Dr. Naveed Aziz, Chief Administrative and Chief Scientific Officer of CGEn. Thank you all, and good morning and good afternoon, wherever you are. I know we have panelists from the West Coast, and thank you for being with us today. The format of the session is that for the first 30 minutes, approximately, we hear from our panelists, and the second half, we will have the question and answer. Please do not use the chat line for your questions, rather use the Q&A box at the bottom of your Zoom screen. And without any further ado, let me ask Dr. Lopez-Correa, the new Executive Director of the project, to start the panel. I believe she's in the job for less than 2 months. Is that right, Dr. Correa? Okay, that's please. The mic is over to you, and please help us leading the way for the panel. Thank you.
Yeah, well, thanks for the introduction, and bonjour à tous. C'est vraiment un plaisir d'être ici aujourd'hui. And yes, I've been less than 2 months in the job, Executive Director of CanCoGen Network, and it's my pleasure to lift the curtain on this initiative and talk to you with the experts on the viral side and the human side. And I'll first give you a short introduction on, for those of you that are not very familiar with Genome Canada. So Genome Canada is the entity that is coordinating this initiative. It's a non-for-profit organization that's working in advancing the use and implementation of genomics in a multi-sector. So we have projects in agriculture, in forestry, in human health, in all different sectors. And we work in very close collaboration with 6 regional centers across Canada. So as Mehrdad said at the beginning, CanCoGen, the Canadian COVID Genomics Initiative, started recently, was funded in April, end of April, and is a national network that is aiming to use genomics to support public health efforts in Canada. So it's an initiative that goes from coast to coast in Canada that is involving scientists, government, public health labs, and private sector. And Genome Canada, the role is really a coordinating role to help all these pieces, put these pieces together. So the funding is $40 million for the viral and for the human side of the project. So we are focusing and aiming to have 150,000 viral genomes and 10,000 human genomes. And so CanCoGen is working, as I say, with many different regional partners, and we're also collaborating internationally. that's a very important element because, as we all know, this virus is not just happening in Canada. It's a pandemic, it's a global issue, it's a global challenge, and that's when international collaborations are even more relevant. So just a small word on the viral side. So we know the SARS-CoV-2, which is becoming the most famous name now, is a tiny little virus, 30,000 letters of RNA that is embedded in a proteic cover. And that little tiny virus enter our cells, hijack the system, start to make copies, destroy the cells, and reproduce themselves and go and spread across the planet, as we have seen. And one phenomenon that's happening when the virus is copying and duplicating and replicating is that there are tiny mutations that happen once in a while. So when these mutations happen, basically they're helping us differentiate different types of virus, and they're helping us understand where the virus are located and how they're moving across the planet. And that's why genomics and reading this RNA is so important to help us understand the movement of the virus and basically building what we call a phylogenetic tree or a family tree. So understanding where the virus is moving, and Dr. Brinkman and Dr. Van Domselaar will explain us a little bit more about that. So there are already about 50,000 viral genomes that have been published, but we need many, many more to be able to understand the movements, the behavior, and the capacity of infection and the outbreaks of the virus. And that will be a key element to help us make public health and policy decisions based in our understanding of the virus. On the human side, what we're hoping is to understand why there is so much variability in how we respond to the infection of the virus, why some people are asymptomatic, why some others have very severe symptoms when they have the infection. And there's already evidence that human genetics has a big role to play in how we respond to this virus. So we're going to sequence these genomes of 10,000 individuals, and we're expecting at least 2 outcomes of this. We're expecting to develop biomarkers to help us understand clinical progression, who will be more severely affected as compared to less severely affected. And will help us, the second outcome is to help us develop precision drugs, precision medicines, and really develop the treatment that is the right treatment for the right person with the specific virus. This initiative is led by CGEn with collaboration with Genome Canada. There are 3 platforms, one in Toronto, one in Montreal, one in Vancouver, that will sequence these human genomes. And Dr. Strug and Dr. Naveed Aziz will explain us a little bit of those components. So just to finish, this is an absolutely unique initiative. It's basically one of the few in the world that is putting together the viral and the human in the same initiative, understanding the viral genome and the human genome. And all this is possible because we have really investing 20 years in genomics in Canada and in science. And that really gave us the capacity, the tools, the training, and the understanding to be able to respond in a much faster way. So with this, I think Dr. Brinkman has a slide that will show us with listing some of the collaborators we have. Is a massive effort that really has many different players. So we see the list of partners on the host seq and the virus seq. And we will start here with Dr. Fiona Brinkman explaining us the aspects that will really, how we're taking on this virus seq. So all to you, Dr. Brinkman.
Thank you. It's my pleasure to be here and talking about the virus seq component of this project led by an impressive implementation committee involving Terry Snutch and also Gary Van Domselaar, who will be talking after me. So as mentioned, this is really about sequencing these genomes and primarily about more coordination and sharing of viral sequencing and associated clinical epidemiological data between provinces, because the provinces have tended to work more as silos, since, as many will know, healthcare is a provincial jurisdiction in Canada. So there's really primarily an effort involving more coordination of efforts across provinces, as well as aligning with global sequencing efforts. And a critical need is also capacity building, both now and for the future. So, but as was mentioned a bit, why we care about sequencing the SARS-CoV-2 genomes, that's the virus that causes the COVID-19 disease. This key benefit is being able to better track transmission trends, capitalizing on these lineages of the virus as they evolve, allowing us to better track spread, not just internationally, but also regionally, provincially as well. So just very briefly, as was alluded to, just so you understand this, the idea is that, you have this first virus, for example. Here's a first annotated viral sequence collected from an individual in Wuhan, China in late December 2019. And this is sort of annotated with these different genes in this genome. Over time, this virus transmits through the population and can mutate. On average, this virus mutates about once every 2 weeks. And what happens is, here's an isolate collected in Germany in late January that has acquired 2 mutations relative to the original Wuhan isolate shown here. And we can use these mutations to generate this family tree, as was mentioned, of a sort of relatedness between isolates. And then over time, this virus spreads and diversifies, and the diversity and location information, along with other metadata, basically inform development of the outbreak. And you'll start to see trends. For example, this Canadian isolate shown here, North American are shown here in red. You can see that in this tree that many of these North American isolates came from European origin, for example, according to this tree. This has important policy implications because, for example, you can look at international transmission. This is a tree showing, a tree based on viral sequences. This time the US is colored yellow and the Canadian isolates are colored orange. And as you'll notice here, there's this speckling of different orange isolates in Canada that predominantly seem to be coming from the USA, as shown in yellow, but notably there's been multiple introductions of these Canadian isolates into Canada from global sources. And also we've given back to the US, so to speak, because here's 2 isolates showing here that suggest that they are of Canadian origin because they're nestled amongst a bunch of isolates that are associated with Canada. So it's data like this showing that there's multiple introductions occurring from global sources across borders that has policy implications regarding border closures. But there are other reasons why we sequence SARS-CoV. And another important issue is identifying evolving characteristics that might impact detection methods, clinical outcomes like strain severity, how well it spreads, and even the effectiveness of drugs and vaccines that are being developed. So for example, there was a change, one mutation involving a change from a D form to a G form of this position 614 in this spike protein, that little spiky protein that you often see in pictures of the coronavirus. And this form of the isolate was sort of spreading more, and so it's being studied intensively to see if it actually does involve more transmission, and also was looked at in terms of disease severity. There's not evidence right now that it causes more severe disease. In fact, evolutionary modeling suggests we are not going to move towards more severe disease, but there is selective pressure on this virus to develop more ability to transmit more easily. But we're just getting started. In closing, this project, VirusSeq project, is really just getting going with the operating principles, infrastructure, protocols, is part of this coordinating and getting organized with data across Canada. But one thing I want to emphasize is that this is really forming a key base. Doing all this work is a key base for that has applications for other infectious diseases as we get organized with how we're going to do what we call genomic epidemiology, or using genomics in helping to track infectious diseases of a variety of kinds. And it's really setting up a model for data sharing in general that will be of more broad use. So I'm going to pass this over now, though, to Dr. Gary von Domselaar, who's our heads bioinformatics at the National Microbiology Laboratory, who's going to talk about the public health perspective.
All right, so I have been, well, I'd like to describe a little bit of the public health, Canadian public health system and how it's involved with CanCoGen to actually translate the work that we're doing into public health policy. So, so at the cornerstone of all public health really is, really strong epidemiology, which we use for things like surveillance at a national scale, but also at a regional scale and even at a local scale. I think a lot of Canadians are much more familiar with the concepts of epidemiology than they would have been, say, 6 months ago. But in essence, it comes in, well, the most basic variety, and the one that people are mostly familiar with is referred to within public health circles as shoe leather epidemiology. So this is where we are noticing trends, syndromic trends that suggest that there's been outbreak of disease, and then we follow that up with interviews to gain more information about, well, specifically about the age, the sex, the time, the location, and other relevant details like who you may have been in contact with. This is referred to as, well, descriptive contextual data or metadata. So there. But when we are, in the modern era, we can apply molecular tests to assist in the shoe leather epidemiology to provide better evidence. So we hear all the time about having these, positive cases in Canada. About 110,000 have been reported. And the way that we do that test is a molecular test. So it gives us a positive or negative value that we can use to say, yes, that person is has the infection, or that person, well, there's no laboratory evidence of that infection. And so this is important information that we add to the traditional epidemiology to be able to provide that extra confirmation. Genome sequence data provides an entirely new dimension to our laboratory-based testing, because it can help, well, it allows us not just to tell whether a case is positive or negative, but it can link them together. And so this is something that we are very active in the Canadian public health system in coordinating, especially with CanCoGen. So Fiona mentioned that the public health is multi-jurisdictional in Canada, and so the Canadian Public Health Laboratory Network is a network of public health laboratories throughout Canada that collectively coordinate the laboratory surveillance and outbreak investigation for circulating infectious diseases. And the CPHLN, for short, is its headquarters are at the National Microbiology Laboratory, where I work. And at the National Microbiology Laboratory, we have, well, we've invested pretty heavily in genomics technologies to allow us to be able to apply that extra dimension, to be able to inform disease outbreaks and to perform surveillance. And a big part of that actually has been from investments from Genome Canada. We're very grateful for the investment that Genome Canada has made at the federal level. At the provincial level, there are some laboratories that are capable of doing their own genomic analysis. There are others that, well, they're still working their way up the curve. And a big part of the effort of CanCoGen is to try to build the capacity throughout the entire Canadian public health laboratory network so that they can all get in the game of performing generating that sequence data and performing that analysis. Okay. I think Fiona really, nicely illustrated the major applications for sequencing of the SARS-CoV-2 genome, but here I'm just going to summarize them quickly. It allows us to perform national lab-based genomic surveillance of the outbreak in Canada and also internationally. The data that we generate, remember, we can link related and unrelated cases so we can detect disease clusters in a multi-jurisdictional public health environment through the CPHLN. It also supports our contact tracing efforts so that when you are trying to identify perhaps which community may have spread the outbreak to another community, you perform, or through individuals, you can do the contact tracing, and the whole genome sequence data can help to resolve any ambiguities in the contact tracing data. So Fiona also mentioned that it allows us to monitor important changes in the virus that can impact our testing methods and also may have implications for disease severity and for transmissibility. And then finally, the large-scale application of this of whole genome sequencing allows us really to gain a really unprecedented amount of understanding about the virus and its evolution, how it transmits, and also how it affects the host. So, yeah. In order for us to be able to do this in a way that is useful and to maximize the ability to apply this for policy making, it's really important that we develop and apply really common standards and protocols. So there are about 5 different sequencing centers that are active in generating sequence data for the network, and then we also have a couple of provinces that are contributing. The National Microbiology Laboratory is contributing, and over time we expect all of the public health labs to be able to contribute. So in order for the data that they generate in this distributed environment to be useful, it has to be properly quality, has to have quality assurance and quality control, and that should be consistent across all the participating sequencing centers and public health labs and our healthcare partners. We also want to make sure that the metadata, that contextual descriptive data that is being collected from the nation is also very consistently described and also very usefully described, so that we can make good sense of the data and use it basically to inform public health policy. And it's also important for us to be able to contribute the genome sequence data and that metadata to the public archives. And here we face a bit of a challenge in the same way that the rest of the world does. So there are privacy implications for the data that we collect because they do belong to the individual. And so we have strong protections in place to prevent the identification of an individual from any of the data that we publicly disclose. And so there's a bit of a tension that arises from the importance of the metadata that we are collecting for the national effort and for the international effort, versus the requirement to be able to protect the individuals that we collect from being identified up into the public. And so there's a bit of give and take here. The public archive, the accepted public archive for the deposition of genome sequence data and the metadata is called GISAID. It stands for Global Initiative for Sharing of All Influenza Data, and they've extended it out for SARS-CoV-2 data. This is accurate to the weekend, so there's over 67,000 submissions, and over 94 countries have submitted their genome sequence data and their metadata to the public archive, so that we can globally contribute to our understanding and the analysis and response to COVID-19 outbreak. And so the little chart that I'm showing here on the left shows some of the metadata fields that we collect. Remember, age, sex, location, and time are the really important ones. And there are others. Some are more well supported than others. So we can see at the bottom for date, region, and country and division, which is sort of the specificity of the region that you're in. People, the countries are very faithfully depositing that information. But when it comes to age and sex, there's a little bit more reluctance because we're getting a lot closer to the actual individual, the patient data here. And so there's, there's concerns about if you combine this, this data in a coordinated way that it could be used basically to be able to identify an individual. So we face this challenge here in Canada. We also face it. The world is also facing a similar challenge. Okay, and that's it for me. I think at this point I'll be turning it over to Dr. Strug, who will introduce the host sequencing.
Yeah. Great. Well, thank you for the invitation to share with you the motivation and the progress for the CanCoGen HostSeq project on behalf of the HostSeq Implementation Committee. So basically, why do we want to sequence the host? So we see that individuals who are exposed to the SARS-CoV-2 virus, so the virus that causes COVID-19, have very different outcomes. So some are exposed and don't even become infected, while others become infected, but their disease can range from asymptomatic to severe hospitalization and even death. And disease heterogeneity is really a hallmark of infectious disease, and previous experiences have shown that the genetic differences between individuals contribute to the infection susceptibility, disease severity, and complications. So understanding the genes that determine the physiological and immunological processes underlying clinical experience can inform who will go on to severe disease and death, and that could guide personalized public health recommendations, prioritization of care in hospitals, such as risk stratification, vaccination deployment, clinical trials, and of course, it's fundamental to our understanding of optimal a design of effective therapies and vaccination. So human infection and disease severity are a result of complex interactions between the pathogen and the host, and it's affected by the environment. So it's really important to understand these host and pathogen-specific factors. So many pathogen-specific factors will be informed when the virus sequencing that we just heard about before is paired with that metadata. So for example, does genetic variation in the pathogen impact disease severity or infectivity? Or what role does pathogen dose play? And these are important factors for us to know. And there are also factors that are specific to the host that we've been hearing about. So for example, pre-existing conditions, such as diabetes and hypertension, they provide underlying susceptibility to disease severity. And then there's data on racial disparities that we really need to better understand. But what will really directly benefit therapeutic and vaccine development is the identification of host genetic factors related to the virus-host interaction. For example, genes related to the viral entry into the cell, such as the ACE receptor that we've heard a lot about in the media, or genes related to immune response. So what do we know now about the genetics of COVID-19? So, well, there's been a twin study out of the UK that showed that symptoms of the virus and susceptibility to the virus are shared within families. So example, fever, loss of smell and taste, or just the frequency with which individuals in the same family become infected. Previous infectious disease studies have shown that rare genetic variation that would be seen by sequencing the whole genome can explain some extreme phenotypes, such as why healthy young individuals develop severe disease when they don't have any underlying comorbidities. There are several candidate genes that have been proposed. I talked about one earlier, but so far the data has not been that supportive for these particular candidate genes, although this is of course an ongoing, evolving investigation. So instead of just looking at candidate genes, there was a study looking at common genetic variation across the whole genome in European individuals from Spain and Italy that was published in the New England Journal of Medicine in June. And this study identified 2 genomic regions that are associated with severe respiratory disease compared to population controls. And this included the finding of the ABO blood locus. It was extremely impressive that this study was carried out in such a short period of time with approximately 2,000 cases, and they managed to put together a large sample of population controls as well. These were convenience controls, many of whom were blood donors. So it remains unclear given the study design whether the genes identified are host-specific factors or are related to the host-virus interaction. But these studies highlight several components of host sequencing that we need to consider, such as the utility of sequencing the whole genome rather than just candidate genes or just common variants, the need for large sample sizes in these studies, careful consideration of pathogen-specific and host-specific factors, and the need for genetic studies in populations of non-European ancestry. So this leads us to the HostSeq project that we're here to talk about today. So the goal of the HostSeq project is to generate whole genome sequencing of 10,000 Canadians exposed to SARS-CoV-2 and to deposit the sequence and the clinical data into a data bank that can be used by researchers across Canada and internationally. So this large data set will be used to identify rare and common genetic variation that impacts both susceptibility and disease severity of COVID-19, which can support therapeutic and vaccine development. Now, accomplishing these goals requires a national collaborative effort where we're coming together with many researchers across the country and forming a network of studies of COVID-19 for which the HostSeq project is producing whole genome sequencing. So as of last week, our network includes 11 principal investigators spanning many clinical specialties, as you can see here. These principal investigators are located in 5 different provinces, although their studies span many provinces, with different population studies, such as inpatients and community sampling. And the genome sequencing will complement other biological data that these investigators are collecting and are generating on the same samples for their own investigations. So we really hope that many investigators will join the network. The process requires just simply completing a short intake form, largely to ensure that the PI's study consent is eligible for participation in the data bank, and legal agreements are signed to transfer specimens and data. And ultimately, the researchers will receive the sequencing, and it's also going to be deposited in the HostSeq databank for sharing with others, as we've already heard the importance of sharing across Canada and internationally. Now, given we still do not understand the extent of organ involvement in asymptomatic or mild infections or severe infections, that highlights the need for a national data linkage strategy that we put in place together in coordination with the network to ensure deep clinical information can be obtained in the future on the participants. So with that, I'd like to hand it over to Naveed Aziz to talk a little bit about the policy implications.
Okay, thank you, Lisa. So just like to start off, I don't have any slides. It's just I'd like to start by thanking Mehrdad and the CSPC team for arranging this exciting and informative panel. My colleagues have done a great job in providing great insights into CanCoGen, and Lisa just before me, thank you for the whole SickKids stuff. So I'm just going to very quickly talk a little bit about some of the key implications that we have, key implications of the project on Canadians and on the Canadian policy. So the first one I want to just kind of reiterate is that putting all of this into perspective, reminding everybody that CGEn has been mandated by the federal government to sequence the 10,000 genomes. We have, as Catalina had mentioned earlier, CGEn is pan-Canadian with nodes in Toronto, Montreal, and Vancouver. But most importantly, it's also home to over 200 expert staff that are involved in not only generating the genomic data, but also are internationally recognized and have expertise in analyzing genomic data and other data sets. So in the context of COVID-19, we hope to gain new insights into how the virus affects a human body by comparing the genomes and the associated rich clinical data that we are gathering for people of varying outcomes and clinical presentations of COVID-19. Lisa talked a little bit about the wide range of the disease outcomes that we are seeing. These are people who are getting severely ill, and people who have mild symptoms. Then there are people who are asymptomatic, don't show any symptoms. But then there are also people who've been dubbed as resistors, people we're very sure are exposed to the COVID-19, but don't contract the disease at all, right? So this project has the potential to dramatically improve our understanding of COVID-19, and which in turn will help us identify underlying genomic differences that play a part in the way people react to the virus, and then the manifestation of the disease. Now, another point I'd like to make here is that the added advantage of CanCoGen as a whole is that it will allow for the analysis of the viral genome data in combination with the host genome data, providing a very unique platform for researchers to understand the human response to coronavirus infection, leading to data that will help control the infection spread, but also help with new treatments. Last point in this context is, if you need to look at the Canadian context, it is very important to recognize that while other countries are generating these data sets, for example, UK, US, Europe, these data sets are very specific to their own gene pool. This Canadian data set that we are generating will represent the diversity of the Canadian population. And hence will be a more appropriate for identification of targets, design of new vaccines and drugs, and prediction to the effect on patients. The second point I'd like to make is around policy, as I said in the beginning. So around the effect of host CGEn and CanCoGen as a whole on the national policy. So if you think about it, since the WHO declared COVID-19 a global pandemic, it became very apparent that this potential disease was testing the capacity of governments around the world in many ways. While we're trying to figure out our response to the pandemic at breakneck speeds, which includes the effort towards, for example, a possible vaccine, for now, policy interventions remain the critical means for governments for managing, sometimes successfully, sometimes not, to save lives and to maintain public confidence. Now, these policies, among other things, are underpinned by scientific evidence and real-world data. It is the scientific evidence, for example, that the kind will be produced by CanCoGen that will provide us with insights and ability to triage the community and high and low-risk population. If you look at history, the concept of preventative medicine took root in the wake of the Spanish flu, right? So the push for prevention became the foundation for the field of epidemiology, for example, where the concept was very simple. What if measures are taken to prevent the onset of disease before it became a pandemic? So in case of COVID-19, it will be understanding of the evolution of the virus and the control through tracking and the ability to focus on members of the population that have a higher vulnerability to the disease. Again, these policy interventions must be rooted in evidence of science, evidence-based science and research. In terms of national policy, another important point that I would like to highlight here is, while we figure out the national response to the pandemic, we cannot, and I can't stress this enough, we cannot underestimate the policy implication for underrepresented communities, including the indigenous population and other remote communities. We have to make sure that any response to this deadly disease provide equitable opportunities for all Canadians. For example, when we do have a vaccine, not only it needs to be effective again for all Canadians, but also what's the yardstick that we will use that will provide insights into the hierarchy of who gets access to the remedies first. How do we make sure that the science-based evidence that we generate includes representation from all communities? For example, as I said, the indigenous communities. Recognizing this, we have spent considerable time and effort in working with various researchers and groups who have in-depth knowledge and experience of and understanding of the challenges we face in making sure that all underrepresented communities are part of the solution of our response to COVID-19. So just wrapping up here, if you take this a step further, we have to also consider the longer term implications of the data we generate today. In terms of consent, in terms of privacy, intellectual property, policy around use of information, by for-profit industry, genetic discrimination, and all these things. It is very particularly important in the context of concepts around collaboration and coordination efforts, both globally and between countries, and also locally between provinces and states and federal governments. While we recognize the core value of open science and that good science requires global connections, Canadian researchers and research teams need to also take appropriate steps to strike a balance between in order to mitigate growing concern related to security, cybersecurity, IP leakage, theft, national security. There was some stuff in the news recently that I'm sure you all have heard. So in the context of the government and national funders, they are all urging all Canadian researchers and institutions to practice due diligence and good research hygiene. As a final thought, I suppose I would say that biotechnology, genomics, informatics offer a growing range of approaches to help detect, monitor, diagnose, and treat infection diseases. But appropriate policies are, however, necessary to ensure that the right tools reach the right people at the right time. Thank you.
That was excellent. Thank you so very much, all the panelists, for such an insightful discussion and presentations. I really enjoyed. And as you may be able to see, that we have very engaged audience. Apparently they enjoyed it very well. We have at this point 153 participants in the room, and already so many questions that we are going to read them all, or to the extent that we can and time allows us. But meantime, just before going to the questions, I want to ask my colleagues at CSPC office to share one question survey with the participants, and I want to ask all of you, please, if you don't mind, to answer that question about the sector that you represent and which sector you're coming from. And also, meantime, please check the chat line for information about our next session, which I will announce at the end of the current panel. Okay, so without any further ado, let's go to the questions, and I want to read them for all the panelists. And I appreciate if you could keep your answers short so that we can get to as many questions as we can. And also, please let us know who would like to answer. I would refer perhaps based on some directions that I have, but please feel free to have, if you have additional comment, let me know. So the first question is from an anonymous attendee. What evidence do we currently have that disease severity is linked to human genetics? Maybe Dr. Strug, you want to take that question?
Sure, I'm happy to. So there's certainly lots of evidence from previous studies of infectious disease where they've identified genetic components that are predictive of disease severity. For COVID-19 specifically, there has been the particular heritability study, the study of in the UK of family members, a twin study that shows that the symptoms the disease symptoms of COVID-19 run in families, and that susceptibility itself runs in families. So these are all supporting pieces of evidence of genomics.
Okay, and the next question from Elise Hope. Dr. Lopez-Correa, you alluded to this being part of collaborative international efforts. Would you please share more about these connections, or those connections, Dr. Lopez-Correa?
Yeah, well, thank you, Alice, for the question. And yes, indeed, the power of CanCoGen is its capacity to really connect with other initiatives. So on the viral genome, Gary already expressed how, all this data is already being deposited in an international database, GZET, that is really collecting more than 67,000 now that we have genomes from the viral side. We have, in terms of human and viral, both of them, collaborations that are starting with the UK. We're looking also at collaborations with the US and other regions, because really, again, the power of CanCoGen, the power of the data we're generating is not just the data we will generate, but our ability to connect with other findings, other data internationally. So that's a work in progress, but the initial MOU, I think, that was signed with CanCoGen and was with the UK, but the idea is to expand this with other partners.
Excellent. A question by Brian Corey. Is there any plan within CanCoGen to sequence the host immune response, i.e. the B cell and T cell immune response of infected individuals? Dr. Strug, do you want to take that?
Sure, happy to. So this brings up an important point that the HostSeq project is, providing genome sequence and collaborating with a network of principal investigators across the country who are running their own studies and asking specific questions related to all kinds of aspects of the disease, one being the host immune response. So there are principal investigators that we are collaborating with who are sequencing B cell and T cell looking for the specific immune response of the host. And so the genomic sequence that we provide to them will be very complementary in those efforts.
Thank you. Next question by Fraser. How can CoGen sharing the efforts with the general sharing your efforts with the general public and avoiding silos, or is the focus on working with public health decision makers? Maybe Dr. Lopez-Correa would like to answer.
Yes, thank you. So there are several communication components in CanCoGen. So there's one part that is communicating with the general public, and we have been very active from the beginning in social media and different, there's coverage on the Global Mail, different newspapers to help people understand what this initiative is about. So that's a big part of what Genome Canada is trying to do with all the partners on the network, CanCoGen network. And there is the other piece to your question, which is the public health authorities. And of course, the whole objective of CanCoGen is not to generate a series of data, put them on a cloud and say, yeah, we got it. I think really we all know, and with the examples we show, the real objective is to connect this data to with public health officials, with policymakers, with individuals, organizations, companies that are doing drug development and therapeutics, so that this data is really meaningful, and this data will provide information, in particular for public health officials. So we are part of the work we're doing with CanCoGen is connecting with these different entities to make sure that the data is used and the data serves the full purpose of the network.
Excellent. A very interesting question by Christian Burks. He's asking, was there a similar genomic sequencing initiative in Canada or elsewhere mounted for the 2002-2003 outbreak of SARS-CoV-1? If so, what was learned from the earlier initiative or initiatives, and to how you are learning from what are the lessons learned for you? Maybe Dr. Van Domselaar, you want to take that question?
Yeah, certainly. So there was a genomic sequencing effort for SARS in 2002. Well, interestingly at the time, we did not have the in-house capability within the Canadian Public Health Laboratory Network to be able to perform that sequencing. So we reached out to actually Genome Canada Sequencing Center to assist in sequencing that genome, which we did. Very rapidly, and we're one of the first countries to be able to generate. I think we're the first country to generate the SARS genome. And well, at that time, just sequencing a genome was a momentous achievement, and it has they had the same potential that it has today. But it was more usefully applied back then for things like rational vaccine design, although no vaccine actually was ever developed against SARS. But interestingly, that was it was the recognized lack of the ability to do the whole genome sequencing and analysis within the Canadian Public Health Laboratory Network that initiated my recruitment to the National Microbiology Lab to establish the Bioinformatics Laboratory alongside my colleague, Dr. Mark Graham, who runs the genome sequencing laboratory there.
Thank you. A question for Dr. Aziz and Elise Hope asking, what efforts is the CanCoGen host sequence project taking to ensure inclusion of genomes from individuals not of European descent?
Yeah, important question. Thanks, Elise, for the question. I talked a little bit about it when I was speaking earlier. The beauty of the Canadian population is that it has representation from a very diverse population, European and non-European. So the idea of having a project like HostSeek 10,000 genomes in Canada in itself captures a diverse population, including non-European descent. But then there are places where and I touched on this earlier, where we know that the representation of certain communities and certain ethnic populations, it will be less in the general population, for example, the indigenous population. For those populations, we are doing targeted efforts, as I said earlier, to make sure that we have inclusion of those populations in the data set. As we gather more and more information on the type of samples we are including into the HostSeek project, we will be assessing the needs for if there is any specific need for a type of population that we need to target in adding to the data set. But as I said earlier, the bottom line is that Canadian population in itself is so diverse, and it's a brilliant data set. In fact, other countries are looking and already talking to us about the diversity in the population and potential access to that diverse data.
Excellent. Yes, Dr. Lopez, you might.
Yes, I just wanted to add a small comment on that. And yeah, we start from a very diverse population, and that, of course, will help HostSeek to get an important diversity. But to ensure equity, diversity, and inclusion in CanCoGen, I think we need to be proactive, and we have been already proactive. And actually, that question, Elise, illustrates very well the power of the enterprise, the genomic enterprise, and the network, because we're already working with Genome BC and with one of the projects that was funded previously called the Silent Genomes Project, that is working with First Nations and really figuring out what is the governance, policy, and ethical aspects on data collection, sample collections with First Nations, and working hand by hand with First Nations and other minorities across Canada. So again, that illustrates that, the power of this initiative is that we are a network, we are working with the genome centers, we are working with the different entities, public health labs, to really get to this diversity we need in this cohort.
Excellent, thank you. I just want to make sure that everybody gets a question, and there is a question for Dr. Brinkman, and please let me know if this question has been already answered, that it's about the targeting 150,000 viral genome with a low mutation rate. Do we expect to see a lot of differences in these genomes? Could you speak to the public health reasons for doing so many?
Yes, thank you. That hasn't been answered yet. What I wanted to bring up is that this is indeed a situation where this genome thankfully isn't mutating very quickly. And so this is a good point. However, what we do find is that these differences are occurring at a level that allows us to sort of track possible international transmission versus community transmission. We can look at identify clusters of cases that have the same sequence. And perhaps most importantly, I think it's really important to keep sequencing these viral samples as they come out over time, because we need to be able to detect when there's going to be a change that might be impacting the ability of a vaccine to function well, a drug to function well, or as had been noted by Dr. Gary von Domselaar and others, this idea of disease severity, or so we could suddenly see maybe a new change occur in the spike protein that would be potentially having an impact on disease severity, and we can look and see if those cases are more severe that have that particular variant versus others. So it is very important that we continue to sequence viral genomes, both the past ones that can help inform how this disease has been transmitting, but also future ones as we sort of see this disease evolve.
Thank you. A question for Dr. Strug. Will you be looking to identify or study antibodies in human populations? to help our understanding of the extent of and risk factors associated with infection?
Right, a great question. So I mean, we ourselves are not studying antibodies, but like I mentioned earlier, that we are putting together a network, and many studies in the network are indeed interested in studying the antibodies, and also to integrate that data with the genome sequencing, which will make for a very powerful analysis to understand what's happening there. We are, however, very interested in collaborating with the immunity task force, because this data bank needs to be comprised of individuals who've experienced the full disease spectrum, including those who are asymptomatic. And serology testing, widespread serology testing, once it's accurate and in the community, will be able to pinpoint those individuals who are indeed asymptomatic and can be included in the data bank to support the research across the full disease spectrum.
Thank you. An interesting question for maybe Dr. Van Domselaar. With the variations, how the vaccine will be uniformly applicable to people?
Oh, right. So that's a good question, and we don't really have a very good answer. So to clarify the question, essentially we develop a vaccine against a certain target, like a surface-exposed protein, like the spike protein, and then we hope that will provide broad protection to the population. But as the virus mutates, it may acquire mutations in that region, and that could inactivate that vaccine, make it ineffective or reduces its protective effect for the people who acquire that variant. Well, the whole genome sequencing allows us to perform what's called rational vaccine design. So some parts are allowed to mutate and some parts are pretty much just not. And the trick is to find a good region of the viral genome that is stable enough to provide long-term protection. It's not meaning mutations occurring in that area, but at the same time is accessible to our immune system so that it can properly neutralize it.
Thank you. A lot of questions remain to be answered, but we will have a small window for one or maybe two more questions. But before that, I want to ask my colleagues at the CSPC office to share the second question about our performance, how we do, and how you enjoyed this session. Please let us know of your analysis and ideas. We would like to know how you think of our sessions. It would be important for us to know how we perform and how we can do better. And also, I just want to mention that the next session will be next week on Monday, July 27th at noon, an interview with Dr. Peter Singer, Special Advisor to the Director General and Assistant Director General of World Health Organization. And the subject or title of this interview is WHO, Canada, and COVID-19. I invite all of you to register and participate in that session as well. And perhaps the last question, and I want to ask the panelists who you would like to take this question. Beyond genome sequencing, single cell studies show that different cell types are infected in different ways, that immune responses to infection varies across patients with varying severities of disease. Is CanCoGen looking to implement single cell and autoimmune studies? Anybody would like to do that, to take that question? And do you want?
Just very quickly, or Catalina, go ahead. You've had your hand up, so go ahead.
Well, no, I just have, CanCoGen is focusing on the viral and the human genomes, but there are many other initiatives. Even Genome Canada is funding some initiatives across Canada, working specifically with the regional genome centers and with labs across and research centers across Canada. And yes, single cell is part of those, is starting. So yeah, Adriana, thanks for the question. We are looking at that, and that will be maybe not as big as CanCoGen, not for 100,000, you know, millions of dollars, but yeah, it is starting. And Naveed, I don't know if you had any other comments on what you're doing on single
No,
cell.
I was just quickly going to say on the multiomics that yes, there are other studies on the metabolomic and the proteomic side that the people are doing that we are connecting with and trying to figure out that the data bank that we develop for the host-seq project, how can that be set up in a way where it can be at a, you know, in the future, in the near future, connected with other databases. So that's in the front of our mind as well, and it's an ongoing effort.
Thank you all. And there is last question by CSPC office. Which region of Canada or perhaps internationally you are attending this session? I really appreciate if you could tell us about that as well. That helps us always to improve our sessions and knowing our audience. And with that, I guess we can call the end of this session. I want to just once again thank you, all the panelists. That was a fantastic session. As you can see, we had a hugely engaged audience. I think we should repeat this perhaps session in months from now, so that we look at what you have achieved with your coordination and projects and kind of report back to the community. Once again, I want to invite you all to look at our website and to see what's happening in the future events. And I want to thank participants for taking their time of their busy schedule and be with us and listening to this session. Thank you all. Thanks Genome Canada for their partnership in this project and this session, and look forward to have other sessions with the panelists in other occasions. Thank you very much.
Thank you. Bye-bye.